Alright, so today we're going to do a crash course, right? All the main points, okay, about biology AS and A level, okay. So we're going to work out on cell structure for today. So here it is, right. So right. So here is the first chapter, cell structure. So you have to know to make temporary preparation of cellular material suitable for viewing the light microscope. So this we won't going to, right, we're just going to brush over the main aspects. Okay, so revision, draw structure, etc. You have to know to draw plan diagram, calculate magnification and actual sizes. So I'm going to show you the formula. Use an eyepiece reticule SH micrometer scale to make measurements, and use average units. So you've got millimeter, micrometer, nanometer, different resolution and magnification. And explain differences between these two with reference to light microscopy and electron microscopy. So let's start. I'll start off with the definition. It's easier to understand. So magnification is simply how many times the image of a specimen observed is in comparison to the actual one. Alright, so you've got examples, so you've got, let's see, okay, like that. So this unit is really important. I mean, this one is really important when you're even like 6 you need to include that. So resolution is simply the ability to distinguish right between two separate points. Right, simply the ability to distinguish between separate points. Okay, need to know all these by heart. Oh, by logic is very easy. In magnification, when you magnify using lenses, etc., you know how many times bigger the image is in comparison to the actual one. The actual one is usually very small, like really tiny, in micrometers, nanometers, etc. But when you do able to see it using a light microscope, which is available in your school laboratory, don't have electron one, which is really expensive. Light microscope. So light microscope is actually nanometers, micrometers, nanometers, millimeters. So you have your usually 30 centimeter clear ruler, alright, which is in centimeter. Yeah, centimeter. Then you have some scaling in it, which is millimeter, and then micrometer, and then so basically from centimeter to millimeter, of course. So just multiply right now. From millimeter to micrometer, you have to multiply 1000, and then yeah, so basically it's easy from here to here. You just have to multiply by, okay, or if you want this, or simply, I mean not simply, this is simply, or if you want this is in detail. Usually from here I have to multiply by 10. Okay, what's next? So magnification is fine, alright, but prokaryotic and eukaryotic. Alright, so prokaryotes is consists of bacteria, usually they are bacteria. Alright, so prokaryotes. So when we talk about prokaryotes, they consist, alright, they, they consist of only one, alright, containing all the necessary, alright, not necessary, but containing, let's say, all enzymes, all enzymes, okay, required, all basic function, okay, required, we can say that, required for basic function. Now we have eukaryote. So eukaryote is us. Okay. Eukaryote, so cell that have a nucleus usually. Okay. So cell that have a nucleus. Let's write it here. Cell that has a nucleus. Okay. So basically they consist of more than one compartment. So they are divided. Okay. They are divided into compartments. And the most prominent one is the nucleus. Okay, so this is all you need to know. So bacteria, it consists only one compartment. Okay, containing all the enzyme required for basic function, and eukaryotes just humans, an animal. Yeah. So yeah, and have centrifuge. Okay, most of the time in MCQs, or not most of the time, you get it right often in MCQs. Right, you have the centrifuge. Okay, centrifugation. Okay, which you need to know. Centrifugation. Okay, this one has to be like that. Alright, centrifugation. So you have to remember the organelles. Number one, you have the nucleus. Alright, so centrifugation, what it does. So it is just a separation organelle. So here we have the heaviest. Alright, alright. Centrifuge is going to be less heavy. The chloroplast, the mitochondrion, then we're going to have the lysosome, the lysosome, and number 5, ribosome, and then of course you get other organelles. So from here to here it's going to be light, okay, or lightest, heaviest, lightest, okay, heaviest, lightest. Okay, so yeah, heaviest, lightest. So here you get a breakdown of all the organelles that you have, the cell structures found in eukaryotic cell, and you have to Now applying the recognizable function, I guess you know all the how you have seen your textbook, all the cell, all the structures. Okay, but still, let's try to um, I'm not going to show you the structures, but we are going to the most important things, right, that you get for exam, size, etc. So, so of course it's not going to be exact or anything. Different textbooks have different interpretation, but it's going to revolve more or less the same. So let's start. Size, size of organelles. So we first go with the nanometers. So nanometers, right? So first we have the cell membrane, which is 3 to 10 nanometers. Second, we have the ribosome. So ribosome, you have two kinds of ribosome, or two types of ribosome. You have 70S, which is 20 nanometers. You have 80S, which is 25 nanometers. Alright. Then we have microtubule, which is around 25 nanometers in diameter. Alright, 25 nanometers in diameter. These are 4 micronanometers. So these what I'm giving you, you can simply just take a whole page, write A4 paper, write size of organelles, and just note all of them down. Okay, try to see. Watch the video, you can do it at the same time, or try to remember it on a board, write it, and see if you remember. And then it's much easier to have actually. Okay, now we're going to look after the micrometers. So then we have micrometers. Okay, so let's start with cell wall, which is 0.1 micrometer to several thickness. So these I have not seen and should be asking it, but still just for general knowledge, or you don't have enough. So just let it. Then you've got lysosome. Lysosome. I mean that mistake for example. Lysosome. Okay, lysosome. So 0.1 to 0.5 micrometers. Okay, you've got centrioles. Centrioles 0.1 micrometers long to, and no, not 2.5 micrometers long, and 0.25. This is a 25, right? 0.10, let me write it well properly. 0.25 nanometers diameter. Number 4 of mitochondria, which is going to be 0.5 to 1.5 micrometer. This is 1.5 diameter, and 3 to 10 long. This part, then you've got nucleolus, So nucleolus 1 to 2. This, and we've got Golgi, which is 1 to 3. Then number 7, you have chloroplast, which is 3 to 10 micrometers. And lastly, the nucleus. That's why we call it the most prominent, because it's the biggest of all. That is, you can easily see it. So here, so you can simply make a table. All right, capital. That's on an A4 paper. All right, so we need to move to next, and these are just important concepts for you to remember. So in the mitochondrion, you have the matrix. In the chloroplast, you have the stroma. So these both are actually cytoplasm, right? These are actually cytoplasm. Okay, so these are actually they are cytoplasm. What's different? I mean, more or less the same. It's not 100% the same. Okay, and you need to know that these two, they actually produce ATP. Okay. Okay, so what you also need to know is about double membrane organelles. Double membrane organelles. So double membrane organelles, you have actually 3 of them. Number 1 is the nucleus. Alright, number 2 is the mitochondrion. Number 3 is the chloroplast. Okay, there it is. You need to remember. And you have now non-membrane bound organelles. So you have simply ribosomes. Okay, you also have centriole, but need to remember ribosomes. What you cannot see with light microscope first, I mean, you have many structures that you actually cannot see. But there you go. The endoplasmic reticulum, ribosomes you can't see, lysosome you cannot see, um, Golgi vesicles, and yeah, all of them. Okay, so these you need to know about them. Okay, so these two, they have circular DNA. Okay, circular DNA, you need to remember about that. So you've got not an animal. So plot what you have in plot, this is what you don't have. So plot, they have chloroplast. What animal does it have? They have plasmodesmata. So these features, plasmodesmata, these features you cannot see in animal cell. Not an animal, so you have centrioles, and you also have the large central vacuole. Okay, and you also have a myeloblast. This has not been mentioned in a book, but I've seen questions asking students what is where I starch. So then I expect them to answer like amyloblast. So it is just where it is just where starch is stored. In case you're wondering, so right. We also have my virus. Okay, plus the fraction etc. of organelles. Okay, so I'll start off with the virus because it's faster to destroy virus one. Okay, so yeah, so the virus is getting around like that. So of course you need to use the detail destroying it of one for revision. Right. So inside you have a genetic material. Inside you have a genetic material which could be either DNA or RNA. Okay, single stranded or double stranded. There you have the protein code. Alright, so here it is simply the protein code. Okay, which is actually has these like that, like round, goes on like that. Actually, you don't have those two layers. I mean, these layers don't have. It's just a guidance. Okay, so let's try to make it like that. Like you have to just like let's try to make it like that so as not to confuse you. Like have it goes on and on like that. I think you already know all this. So and why I did that to show you that one of them is known as a capsomere. Okay, and that's it. And they have a symmetrical shape. They hijack the cell's machinery to make copies of themselves. Okay, they all of them are parasitic. This is the size, 100 nanometers. Okay, and yeah, they have a protein code or a capsid. Then code it like that, a capsomere. Very symmetrical. They are non-cellular. Okay, as they are not able to carry out any of the processes which are characteristic of living things unless they are inside the cell. I mean not a cell, like a living cell. Okay, so cell could be dead as well. A living cell. Okay, where they hijack the cell's machinery, and of course they cause disease in humans. All right. So now let's look at bacteria. So bacteria, what you need to know. Okay. So this is the flagellum. This is sometimes present. This is a right cytoplasm, and this is a layer, the green one capsule. There you've got the ring, got the DNA, got the small 70th ribosome. Alright, and then what you have, this is the genetic material, okay, which is actually the circular DNA. And here is the plasmid. So let's move over here. So of course the cytoplasm contains all the enzyme needed, okay. And ribosome you have it at 70s nucleode, it is a nucleode, of course they don't have a nucleus, so you have a nucleode, okay, here, um, there, right, this is a nucleode, right, where, or you can call it nucleosome. It's the region of the cytoplasm that contains the DNA, and DNA is always circular. They have a cell membrane as well, which is made up of phospholipids and protein. The cell wall, however, it is made up of peptidoglycan, unlike a plant cell, which is made up of cellulose. And the capsule, or the slime layer, which is green in color, as you can see, it is actually a thick polysaccharide, okay, glycolax, therefore period. So they are used for sticking, okay. Of course, this sticky layer, okay, it also protects against desiccation. It's like a layer, okay, protective kind of layer. And you have a flagellum, okay, which is sometimes present. So yeah, this is all for bacteria. I need to remember bacterial prokaryotes. Okay. Now let's see. So I've made short notes. I've searched for it. So yeah. There are short notes for eukaryotic cell structure and function. You will fast. Otherwise, I have to note it down. My pen, stylus. Okay, one by one. So let's start. So cell surface membrane, which is actually 3 to 10 in nanometer. I mean not nanometer. I mean nanometer. 3 to 10 nanometer. Okay, it's the structure of cell membrane. The structure looks static. The phospholipid and protein forming the bilayer. Cell surface membrane. So all the cells are surrounded by cell surface membrane, which controls the exchange of material between internal environment and the external, that is in and out of the cell. Okay, it is described as partially permeable. So if you happen to have a cell wall, it is fully permeable. This one is partially permeable. Okay, the cell membrane is formed from a phospholipid bilayer. Okay, as I said, the phospholipid bilayer, spanning in diameter around 10 nanometers. So here you have the size, every to 10 or 10 strands. The cell wall, so here is the structure. The cell wall is freely permeable to most substances, unlike the plasma membrane. That's why we said partially permeable. Read this. Partially permeable. It's very important to remember. Like I should learn all this in form 3 or level, etc. So cell walls are formed outside of the cell wall and offer structural support to the cell. Alright, so should not limit yourself. You're told that cell walls only made for plants by of cellulose, okay, just in peptidoglycan in most bacterial cell as well. So narrow thread of cytoplasm surrounded by cell membrane. Cool. Alright, so all this you don't need to know, but I have it there already. Plasmodes, metaconect, cytoplasm of neighboring cells. Right. So the nucleus contains chromatid, okay, usually at least one or more nucleus stain observed. Mitochondria, so this is the powerhouse of the cell. This is where ATP is produced. You have both aerobic and anaerobic respiration. If you're going for A-level biology, you're going to have a chapter called respiration where you're going to learn all this in detail. So a single mitochondrion shown, the inner layer has protein complexes vital for the later stages. The site of anaerobic respiration within a eukaryote, surrounded by a double membrane. How I said earlier, you need to remember. With the inner layer folded to form crystal matrix form. Okay, mother crystal contain enzyme needed for aerobic respiration producing ATP. So all these details you don't need to know. As such, of course, you need to know that where respiration occurs, where ATP, etc. is produced. But you don't need to know that contains all the enzyme needed, etc. That is good to know. Small circular pieces of DNA, mitochondrial DNA, and ribosome also found in the matrix, needed for replication. Chloroplasts are found in the green part of the plant. Alright, so the green color, okay, as you can see, most plants are green is due to the photosynthetic pigment, chlorophyll. So larger than mitochondria, also surrounded by double membrane organelle. So of course we've seen the sizes. Okay, so larger than mitochondria, surrounded by double membrane again. Membrane compartments or thylakoids containing chlorophyll stacked to form structures called grana, which are joined together by lamellae. Chloroplasts on the site of photosynthesis, you have both line independent and light independent stages. Calvin cycle, etc. You're going to learn this in detail in photosynthesis A level. Right, it also contains small circular pieces of DNA and ribosome used to synthesize protein needed in chloroplast for replication and photosynthesis. So you also need to know, I guess you know already, that mitochondria and chloroplasts have evolved. Okay, they were they are thought to be prokaryotes before. Okay, so that's why you can see that they have 70s ribosome. It has something mentioned here, but of course you know it, and you need to know. So ribosome are formed in the nucleolus. Okay, this is a function of nucleolus itself. Alright, its function is to produce um, RNA, ribosomal RNA. Okay, are composed of equal amounts of RNA protein. I've talked about that. Okay, RNA is made up of a ribosome are made up of RNA and protein. So they are found freely in the cytoplasm of a cell or part as endoplasmic reticulum. It is actually studded with endoplasmic reticulum. Each ribosome is a complex of ribosomal RNA and proteins. You have ATS, you have 7TS. Okay, and of course the ATS has a subunit larger part and smaller part. The large part is 60S and 40S subunits, and 70S more. So the 60S, 40S, 50S, and 30S. Don't need to know or remember. Alright, you just need to know that it has a large compartment, a large subunit, and a smaller subunit. How it works, again, you're going to learn in nucleic acid if you haven't already. So yeah, 70S are found in prokaryotes, 80S are found in eukaryotes, and have a larger subunit and a smaller subunit. Okay, that's site of translation, protein synthesis. And the plasmid reticulum, we have rough and smooth. Rough is studded with ribosome, or surface covered in ribosome, and these ATS form from continuous cell membrane continuous with the nuclear envelope. Processes protein made by ribosomes. They made protein, they can even transport the protein and membrane production. right? Synthesis, folding, and modification of proteins. Smooth endoplasmic reticulum, it does not have ribosome on the surface, that's why we call it smooth. It's easy to remember, smooth. It has a smooth layer, it's not studded with ribosome. Rough is rough because it has many ribosome or even ribosome on it, okay? We have a lot of. It's rough, involving the production, processing, and storage of lipoprotein, carbohydrates, and steroids. Alright, you even have hormones, okay, and detoxification of drugs and poisons. Golgi apparatus or Golgi complex. There are flattened sacs or membranes similar to the smooth endoplasmic reticulum. Okay, it modifies protein and packages them into vesicle or lysosome. So one example of modification is that, for example, the protein comes from the endoplasmic reticulum. All right, rough endoplasmic reticulum. You can add sugars to it. All right, carbohydrates is added, or you modify it to remove some of the protein. I don't know. Okay, then the vesicle blood off. Okay, now transported through the vesicles. Right, so there is a lot different types of modification. You can just think of it adding sugars, okay, adding lipids. Okay, so it receive both proteins and lipids received from the smooth and rough ear, both. So large permanent vacuole. There are sac implant cells surrounded by tunnelblast, selectively permeable membrane, vacuoles not animal. So these contain tannins, okay, tannins from which you get tea. Okay, it has all the waste products as well. Vesicle, they are membrane valves, okay, for transport and storage, as I said. They build up from the Golgi apparatus. Even it is involved in the process of, they can even fuse with the membrane, okay, cell surface membrane, to um, for exocytosis, endocytosis, etc. Okay. Centriole, hollow fibers made up of microtubule. The centriole at the right angles to each other form a centrosome, which organizes spindle fibers during cell division. This again you're going to learn in the chapter mitosis. Okay. Not found in flowering plants. Okay, this is highly debatable. Okay, but the scientists have been looking after it, and now it has been found. As I said, it's not found in plants. I have noted it in the whiteboard here. Okay, not found in plants. We have there. Okay, remember, centriole is not found in plants, and do not confuse with centriole, centrosome. What do we have more? Centromere, etc. Microtubule makes up the cytoskeleton of the cell, about 25 nanometer in diameter, made of alpha and beta tubulin combined to form dimers. The dimers and then join into profilaments. 13 profilaments in a cylinder make a microtubule. The cytoskeleton is used to provide support and movement of the cell. So yeah, microvilli, cell membrane protection that increases surface area for absorption. So you have it in small intestine, etc. And many more places. So you have many in the cell. There are MCQs questions based on that. Cilia, there are hair-like projection made from microtubule. So cilia, you have it in our gas exchange system, in our trachea. Okay, allows the movement of substances. So it's like, yeah, it's going to move in a synchronized manner. All right, and move mucus out to be coughed out or swallowed. Flagella, similar structure to cilia made up of longer microtubule, contract to provide cell movement, for example, sperm cell. Okay, so it's like it is like in the tail, alright? So I think we are done. Alright, we've done most of them. Micrograph. Alright, so we've missed this part. So you need to know that prokaryotic cell is found in found in a typical bacterium. Okay, they are unicellular 1 to 5 micrometer diameter peptide like an cell wall, circular DNA, 70s ribosome, abscess of organelle, and abscess. So this is just about prokaryote. Okay, and we need to know that prokaryotes are bacteria. Just think of bacteria when you ask about prokaryotes. So you get rid of larger cells. Okay, they are multicellular, unlike unicellular. Unlike the bacteria, which is unicellular, generally 1 to 5 micrometer in diameter, the eukaryotic cell is actually 10 to 100 micrometer. Yeah, they have bacteria have peptidoglycan cell wall, eukaryotes have cellular cell wall, circular DNA, linear DNA for eukaryotic cell, 70s ribosome for prokaryotes, and ATS for eukaryotes, and absence of organelles surrounded by double membrane. Eukaryotes have actually many organelles surrounded by double membrane, three of them: nucleus, mitochondrion, and chloroplast. Okay, so you compare the structures. Say that all viruses are non-cellular, we've seen either DNA, RNA, capsid, metaproteins. They have an envelope of phospholipid, so we've not seen phospholipid, we've seen here's a virus, we've seen of capsomere, protein code. Okay, so if you have any kind of question, you ask in the comments. Hopefully you like the explanation, you can understand something extra. Alright, and there you go, see you in our next video.
Subscribe to our YouTube channel TSR Academy, like, share and follow to excel.